So Meselson-Stahl experiment, what were the different hypotheses? So you have a double-stranded molecule of DNA. We know it's going to turn into 2. So we have semi-conservative replication. We've got conservative replication. And there was another one which we didn't talk about in class, distributed replication. We can just focus on those two. So conservative versus semi-conservative. So let's assume that conservative replication, we're going to show the new DNA strand in red. What would this molecule of DNA at the top look like after conservative replication? Both strands get replicated, so you wind up having one new double-stranded DNA molecule and the original. So that would be the outcome of conservative replication. It's conservative because it's conserving what this original molecule looks like. You start with one double-stranded molecule of DNA, and it in its entirety is still left. It's conserved after the first round of replication. So what does it look like after semi-conservative replication? What do we know? How do we know DNA actually replicates? What's the first step of DNA replication? You have this molecule at the top, double-stranded DNA. What's step 1? What's the first step? Yeah, you separate the two strands of DNA. Okay. And then semi-conservative replication says that each of these two single-stranded molecules of DNA serve as the templates to synthesize the other strand. So it's semi-conservative because the original black nucleotides, the original molecule is still there. It's just now it's part of two separate double-stranded molecules of DNA after synthesis. So in both of these cases, conservative and semi-conservative, the outcome is the same. You still go from one molecule to two molecules of DNA, two double helices. But there's an essential difference. And so the Meselson-Stahl experiment was to figure out which of these two circled outcomes is the case. Do you get one DNA molecule where both strands have just been synthesized? That's conservative replication. Or do you get two molecules of DNA where exactly half of the nucleotides are new? One strand of the two. So how did Meselson and Stahl distinguish which was new DNA and which was old DNA? Robert? Heavy nitrogen. So what's shown up here in red, the newly incorporated nucleotides contained an isotope of nitrogen that's heavier than normal nitrogen. So we're going to say any red nucleotides involve heavy nitrogen. So what's the expectation then? How do you distinguish outcome 1, conservative replication, from outcome 2, semi-conservative replication? How do you separate things that have different weights? So in this case, Meselson and Stahl centrifuged the DNA. The DNA's different masses, it should centrifuge at different speeds. Sediment segregate at different speeds. So of these three molecules, I'll call them A, all red, B, all black, and C, half red, half black. Which one's heaviest? A, right? B is lightest. It has absolutely no heavy nitrogen incorporated. And then the molecules that are labeled C here, half red, half black, would be somewhere in between, medium. intermediate. So the prediction from the Meselson-Stahl experiment is that when you centrifuge these in a centrifuge tube, if conservative replication is happening, here's a centrifuge tube, you're going to see two bands of DNA that have migrated different distances through the liquid in the tube, the heavier band and the lighter band. Those are bands A and B, respectively. The heaviest band is at the bottom. The lighter band won't have centrifuged as far as this tube is spun in a centrifuge. So what does this exact same tube look like if semi-conservative replication is happening? What do we observe? Where do I put band C? Are there one or two bands? They're both the same molecular weight. They're both each of these molecules is half red, half black. So all of the DNA should be the same molecular weight. So it's all going to sediment at one rate. It's going to move through the liquid in the centrifuge at one speed. So we see one band. Where is that band relative to B and A? It's in between. So you'd expect it at an intermediate weight. So a half red, half black molecule is going to be somewhere in between where you would see an entirely black, light DNA molecule and an entirely red, heavy DNA molecule. And that's exactly what Meselson and Stahl observed after one round of DNA replication. Instead of seeing two bands that corresponded to heavy DNA and light DNA, they saw one band that was an intermediate molecular weight.
So for the first generation, they didn't see any light or heavy?
Right. So in the original bacteria, they only see light DNA because they haven't introduced the heavy nitrogen label yet. After the first generation of replication, they start to see that heavy nitrogen incorporated into molecules.
If it was conservative, would the light and heavy bands stay there throughout multiple generations?
So what happens, yes, what happens over multiple generations of conservative replication? So you've got a light band, you've got a heavy band. So we can look at generations. After the first generation, you get one light band, one heavy band. So we've got two molecules of DNA. That's after the first round of replication. We've gone from one double-stranded molecule to two. So what does this tube look like after an additional generation of replication? Each of these molecules gets used to replicate, if this is conservative. This is not the way that DNA replication works. But you could imagine I would ask a question like this. What would this tube look like after the second generation? Do we still have any light chromosomes? So this one light chromosome is still present. So we'd still see a light band. That was the original DNA molecule, just one. It was there after the first generation of replication. It's used to make another copy. So the original stays around. And in conservative replication, there's also a copy made from that original molecule. What about the heavy molecule in generation 1? This one. What's going to happen to it? Does it get used to replicate a chromosome as well? Yeah, it's a chromosome. It's going to get replicated. So it's going to be heavy itself, so it sticks around in the next generation. So it adds a little bit more thickness thickness to that band in generation 2, plus that heavy band gets replicated. So we have 4 molecules of DNA. That's replication once from 1 copy to 2. That's here. We've got another round of replication, synthesis. It takes us from 2 molecules to 4. But all that's happening is we're getting more and more heavy molecules added. So that one original DNA molecule stays around, and it keeps getting used as a template to synthesize more heavy molecules.
Isn't the original molecule with the heavy band? I thought the parent molecule would be heavy and the
So right. So the question is the original molecule heavy or light? In Meselson and Stahl's experiment, unless I'm mistaken, the first round of synthesis occurred in heavy. So if we went back to generation 0, which you can tell me whether or not we can or can't do, this is before adding heavy nitrogen. All of the DNA, there's one light band, right? Unlabeled, no heavy nitrogen, just a normal double-stranded piece of DNA. And then after that first generation of replication, that original band is still there, plus a heavy copy is made from it. So the original molecule that was the first piece of DNA that was synthesized in the presence of the label, red, heavy nitrogen, sticks around generation to generation to generation. It keeps getting used as a template to make more copies of DNA. But every additional copy gets made in the presence of heavy nucleotides, and they're red in this case. So after an infinite number of generations, you still have that single molecule of original DNA left, same light weight. And then you've got basically a huge smear of an infinite number, after infinite generations of heavy copies.
If this was semi-conservative, would the intermediate kind of like line up and then the light band would be the majority?
Okay, so what would the outcome be over multiple generations if semi-conservative, which is what really happens, occurs? What's the ultimate outcome? So we start with that same generation 0 light band. What do we see in semi-conservative again? First generation, that one light band is split in half. Each of those template single strands is a template for making another strand. So we get 2 half-heavy bands, each of which is 1 strand of black and 1 strand of red. Will we ever regain any light strands? Pure, totally light. Once you have DNA molecules that have 1 strand that's black and 1 strand that's red, is there any way to go back to black, black? No. Because every new piece of DNA that's synthesized is going to be what color? In the presence of heavy nitrogen, red is the label. So every additional piece of DNA we're going to drop here is going to be at least half red. And you can go back to the lecture that we covered this material in, and it shows Meselson and Stahl's original data and the band intensities and molecular weights. So you can compare to this. So we're going to keep having half-heavy bands. In the second generation, what molecule shows up for the first time ever in the second generation? completely red. So when this red single strand gets used as a template to make another copy, second strand, then you've got for the first time in the second generation an all red, all heavy DNA molecule. So eventually, you wind up with the same result. That over time, you still only have two original black molecules, or strands rather. You started with two black strands, unlabeled, original molecules. You still wind up, after infinite generations, with those same two strands, each of which is part of a black-red duplex that are half heavy. Everything else that's been synthesized by the fact that it's being synthesized in the presence of heavy nitrogen makes that huge, flopping, fat, all red band. It's heavy. So what distinguishes the difference between conservative and semi-conservative? What's different between this drawing and the one on the previous slide? What proved Meselson-Stahl's point? What band was left at infinite generations in conservative replication? Was it half heavy? No, it was light. It's on the previous slide. So if that had shown up, if there had still been a light band, that would suggest that conservative replication, those two strands, the original two strands of DNA, are always base paired with each other after replication. That would mean conservative replication. But instead, Meselson and Stahl saw, after numerous generations, that half-heavy band was still there, and that suggested semi-conservative replication.
How come in their image though, that the light band darkens at generation, like at generation 2.5 is black? That's what's throwing me off.
So what does it mean to be at generation 2.5? So the question is what happens to their original data at 2.5 generations? What do we expect at 2.5 generations? That's kind of weird. Which date was that?
17.
Is that like during replication?
So it could have been during replication. Let me pop this up on the screen so that you all can see what we're talking about. Okay, so the concern is, you said 2.5 generations in. So this one. So why is that band darker than then?
Well, I thought on that when you were explaining, you said that the heavy would appear and the light would disappear, but.
So this is a different, so they did their experiment in a different way than I was just describing. So don't memorize patterns of darkness. Think about what the process is that's happening through DNA replication. So it depends on how you set up the experiment, what you see. So in Meselson-Stahl's experiment, how long did they label the DNA for? How many generations of bacterial growth were in the presence of heavy nitrogen? What does it look like up here? How long did an all heavy DNA strand stick around for? How many generations?
One.
Yeah, just one. So they grew the bacteria in heavy nitrogen for one generation, and they took away that label, the heavy nitrogen, and then they let the bacteria grow in the absence of heavy nitrogen. And so that's why you see that heavy band disappear and the light bands reappear. If you do it differently, if you keep growing the bacteria in heavy nitrogen, that's what happens that we just discussed. Every additional molecule of DNA that gets replicated in the presence of heavy nitrogen stays all heavy. So there are two different ways to do the experiment. Meselson-Stahl did it the way we just discussed. They grew the bacteria initially in heavy nitrogen, and then they stopped feeding the bacteria heavy nitrogen once they started the experiment. The way I described earlier, you feed the bacteria the heavy nitrogen through the whole experiment, infinite generations, and you see a different pattern. Both of those types of experimental approaches, and one of the things that I'm hoping that you'll be able to do, if not on the midterm, then certainly exam, that is, for the final, is to propose an experiment to test a hypothesis. And if this was the type of question I was asking, then either of those would be fine, because both of these types of experiments prove the point. They both test the hypothesis, and there's a difference in outcome depending on whether or not conservative or semi-conservative replication occurs. Nice to hear the birds singing again. What else can we do?
Can we go over independent assortment?
Independent assortment.
How to design rounded trace with the trace of combined.
So independent assortment during segregation. Okay. So independent assortment. Setting up Punnett squares, combining traits. So what is true if two traits independently assort? What do we know about the genetic distances between those traits? They're unlinked. It's 50 centimorgans apart. So Mendel's second law, independent assortment, is only true for, it's technically only true for genes that are 50 centimorgans apart. This could mean one of two things. How would...
They're at the ends, they're on different chromosomes.
Right. So this can happen in one of two different cases. Either genes A and B are at the opposite ends of the same chromosome, or they're on separate chromosomes. So here's two copies of chromosome 1, for example. Or genes A and B could be on two separate chromosomes. In either case, you get unlinked, essentially unlinked, independently assorting genes, A and B. Then you've also got, by contrast, you've got linkage. What's the most extreme form of linkage?
0 centimeters.
They're physically right next to each other, and another one of you just said they're 0 centimorgans apart. That means exactly the same thing. So you could have the big A and the little a genes there, and you could have the little big B and little b genes right next door, 0 centimorgans. There's absolutely no chance that recombination, physical breakage of the chromosomes in exchange during meiosis can occur between those two genes. Is there ever a 0 centimorgan distance, or is that just sort of a fictional extreme case? There's probably always some probability that a recombination event occurs between two genes, even if they're right next to each other. But we'll say 0 centimorgans. So in which of these cases do you think it's more likely that you can combine the trait of big A with the trait of little b? So let's say big A is flower area, A for area, dominant, so capital A gives you a bigger flower area. And trait B gives you, what, number of branches. With little b being homozygous, giving you fewer branches. So if you wanted to make an organism that has big flowers and very few branches, what genotypes would that individual have? What are the possible genotypes? What do you have to have at the A locus to make big flowers? At least one, what was that? At least one capital A. So either homozygous big A or heterozygous. What do we need if we want to make an organism that has very few branches? What's that genotype? Homozygous little b. So either of these two genotypes, big A with little b, this is for linkage, a big A with a little b over a big A with a little b, or a big A with a little b and a little a with a little b, would make that phenotype. Big flowers, few branches. Because A is dominant, makes big flowers, B is recessive, makes few branches when it's recessive. Can you actually make that genotype? Given the drawing in the bottom here. 0 centimorgans apart. What haplotype is essential to make either of these two genotypes? What haplotype do both of these genotypes share? What? A little, so both this genotype is all big A with little b, right? These are linked genes. What do we have to work with? We've got big A with big B, that's this haplotype, and we've got a little b haplotype. So if they're 0 centimorgans apart, the A and B genes, are we ever going to see a big A with a little b or a little a with a big B after recombination? 0 centimorgans means 0 recombination between those two genes, which means that we're always only going to ever see big A's with big B's and little a's with little b's. So there's no way in hell to get any of those haplotypes, a big A with a little b, when those two genes are 50 centimorgans, when those two genes are 0 centimorgans apart, when they're perfectly linked. You're never going to get that big A with a little b. So you can't make that genotype, you can't make that genotype. So there's no way to make that combination of traits in this case. And that's because these genes are not independently assorted, they're linked. The gametes will never carry a big A with a little b. So how does independent assortment help us? So focus on this example to hopefully directly address your question. here we expect the genes to independently assort when the two genes are on totally separate chromosomes. So what's the process that allows that to happen? How can we now make a big A with a little b? Two different chromosomes, we've got a dihybrid, heterozygous. Big A over little b, big B over little b. What symbol do I put in between those? When the genes are unlinked, they're separated by a semicolon, right? So that's the genotype of this individual. Big A over little a, semicolon, big B over little b. Okay, how does independent assortment work? Inside the cell, what causes Mendel's second law to occur? We've got a diploid cell. We have to do meiosis for independent assortment to happen. So what's the first step? Start with a diploid cell. What happens right before meiosis? Synthesis. So after synthesis, you get big A and its copy, little a and its copy. Yeah, bless you. Through semi-conservative replication, I might add. You get two copies of the chromosome that have little b. So you got two tetrads. Tetrad 1 and tetrad 2. Now what? We've done synthesis. What do we do right before meiosis 1 happens? So something's going to segregate. We're going to have the first meiotic division. So that one cell in red is going to create two cells. We're going to go from 8 total molecules, n equals 8. There are 8 molecules of DNA drawn up there, 8 vertical lines, to 2 cells that have n equals 4. We divide the DNA in half. Start with one cell with 8 copies. You get 2 cells with 4 sticks, 4 double helices. In this case, this is not yet independent assortment. We have to figure this out. Which letters do I write in which cell? So I'll start you off. We're just going to assume arbitrarily that the big A's went over here. So what's in the other daughter cell? It's got to be the little a's. Because we have to ensure that all of the cells have the same number of each chromosome. So the chromosome that has gene A, you better be sure if it's got 2 copies in one cell, it's going to have 2 copies in the other daughter cell. This is independent assortment now. Which of those cells do I put the 2 capital B's in? The cell on the left or the cell on the right?
Right.
Okay, so I heard right. Which means the little b's have to go over here. Why did the little b's go on the right? Because she wanted them to. So the choice of which of those cells you put the capital B's and the little b's in is totally arbitrary. It's just as likely at meiosis. And again, where you put the A's is arbitrary, but now what's the other opportunity that we have? How else could we segregate those B's? Yeah, big B's on the left, little b's on the right. And those are equally likely. They both happen during meiosis, and they occur in a 1-to-1 frequency. That is, what happens on the left side happens exactly as equally, as frequently, as the division that happens, the orientation that happens on the right. Sorry, I should write it like this. 1, 2, 1. So this arrangement with the little a's and the big A's and the little b's, and the little a's with the big B's occurs just as frequently as the big A's with the big B's and the little a's with the little b's. So now, are we making gametes that have a big A, little b haplotype? Which one of these 4 daughter cells has the type of haplotype we were looking for when we make the big flowered, few branched trees? This one right here, number 1. After the second division, that cell is going to make 2 daughter cells, each of which have a big A and a little b. And that's independent assortment. The fact that you see all 4 possible combinations. You get big A's with big B's with little b's, little a's with big B's, and little a's with little b's. That's recombination. And that's what independent assortment does for us. So it's all about how chromosomes independently line up during meiosis. You get all the possible combinations of haplotypes, all 4, when you're dealing with 2 genes.
So concerning the question in the study guide,
Yep.
you said when they're on the opposite ends of the same chromosome, they act as if they're unlinked. Is it equally as likely for recombination?
Okay, so follow-up question up here is, given one of the questions on the study guide, how does this, when genes are on separate chromosomes, differ from what happens when the 2 genes are at the opposite ends of the same chromosome? In this case, this was this previous slide, looking at independent assortment, genes on separate chromosomes. This is also technically called recombination. This just doesn't involve, this is not the type of recombination that involves physical breakage of chromosomes. This is recombination equals independent assortment. Recombination, new combinations of alleles that have never been seen before. So this is not physical recombination, this is genetic recombination. It's okay, don't worry about it. So physical recombination never happens between genes that are on separate chromosomes. Those physical breaks so essentially the question is, why are they also acting like they're unlinked even though they're on the same chromosome? If we go back to that tetrad stage, let's assume we have a tetrad that looks like that. So when genes are on the same chromosome, we know that every tetrad has a recombination event happen intrachromosomal. That is, the DNA breaks, there's crossovers. So if I'm going to put a crossover somewhere here, between sister chromatids A and sister chromatids, I shouldn't use A, let's call them X and Y. We've got a pair of sister chromatids I'm going to call X, and the pair I'm going to call Y. If you can have a crossover between X and Y, where is it going to happen relative to these 2 genes? Above, below, or in between? Yes, I know you hate me asking stupid questions. You know there's that thing about there is no thing as a stupid question? Sometimes it's the ones that the professors ask. So yeah, in this case, in the case where the genes are at the opposite ends of the chromosome, recombination has to occur between them. So recombination always occurs in every tetrad. It doesn't matter which pair, as long as it involves one of the 2 sister chromatids X and one of the 2 involved that carry sister chromatids Y, the other haplotype. We've got exchange involving the big A, big B chromosome with the little a, little b chromosome, in other words. So after this crossover is resolved, XO crossover resolved, what do these chromosomes look like? What does this first chromosome look like? What's its haplotype after crossover? What alleles does it carry? big A and big B. It wasn't involved in a crossover, so it stays the same. Where's the other chromosome that stays the same? So the third one from the left, this little a, little b wasn't involved in crossing over, so it is little a, little b. Okay, what are the haplotypes of the two recombinant chromosomes? We've got a chromosome that's got the big A, and then there was a crossover, and it picks up the little b allele. So now we've got a big A, little b, and the other chromosome that was involved in the crossover starts with a little a, and then it connects physically to the bottom part of the chromosome, the second chromatid. So that's a little a with a big B. So you get one big A, big B, one big A, little b, one little a, big B, one little a, little b. What does that look like? It's like what we just did on the previous slide with independent assortment. When genes are on separate chromosomes, you wind up getting the 4 different haplotypes produced in equal ratios. In this case, when genes are at the opposite ends of the same chromosome, you still get that 1 to 1 to 1 to 1 ratio of the 4 different haplotypes. So when genes are unlinked, that's what you expect. They're at opposite ends of the same chromosome, they act like they're unlinked, even though they're physically associated with each other. Or when genes are independently assorted, separate chromosomes. What happens when genes start to get closer and closer together? What would you calculate this genetic distance as? If you have an equal number of we don't know which are the parental and which are the recombinant types. But you have to have, let's just arbitrarily say that the capital and the lowercase letters are parental types, and the ones with mixed upper and lowercase are recombinant types. What would be the genetic distance you would calculate in just this one meiosis? You've got 50. You've got 2 recombinants, R plus R. You've got 2 recombinant chromosomes over all of the chromosomes you looked at, 4. Which is 2 over 4, or recombination fraction of 0.5. So both when genes are on separate chromosomes or they're opposite ends of the same chromosome, you calculate a 50 centimorgan map distance between them. They independently assort. As genes start getting closer and closer together, which direction does this ratio move? When genes get closer and closer together, recombination fraction goes smaller. So do you see fewer parental types or fewer recombinant types to make this ratio get smaller? So if we're going to diagram something where genetic distance is smaller, we want this fraction to be smaller, which means we want the numerator to be smaller, which means we have fewer recombinants. So you see, when you observe fewer recombinant types, you get a smaller recombination fraction. The genetic distance between the genes becomes smaller. Recombination happens less frequently between the genes. And the limit to that is you see 0 recombinants, map distance is 0. Genes are perfectly linked.
So will recombination most likely happen to the genes that are on separate chromosomes than for the other?
Right. So the question is, it's regarding recombination on genes on different chromosomes versus genes on the same chromosome. So I haven't explicitly discussed this in class. I'm glad you asked. Like here, only happen within a tetrad. So that's chromosomes that are the same homologous chromosomes. So you never get physical breakage and physical recombination of genes that are on separate chromosomes. one more question if somebody has one. Sir?
Recombination can happen on both chromosomes, right? Not just the one.
Correct. So where else could we have drawn recombination in this tetrad?
First and third.
So you could have involved the first and the third sister chromatids shown up here. Could have been the second and the third,
First and fourth.
and the first and the fourth. And all of those would give recombinant types. Now, close your ears if you don't want to hear something that's going to confuse you. I would never do that to you. Recombination can occur between, I'll do this in some fancy color. Let's see, what color do I want? Pink? Recombination could also occur there. You've closed your ears, right? Okay. So recombination can happen between the third and the fourth sister chromatids. But that doesn't make new combinations of haplotypes. If you don't want to know that, forget you just heard it. But this is truth. I'm telling you the truth about genetics. Not how we simplify genetics for classes. These sorts of recombination events can happen as well. We just don't see them because there are no new combinations of haplotypes produced. You still have little a connected to little b and little a connected to little b. Don't worry about it. So, if you've got one quick question, come on up, ask me right now. Otherwise, like I said, I've got an hour and a half of off.